Okay, I think we should be recording now. Yes. So this afternoon is our eighth session for chemistry seminar. And today we will be learning about molecular information and synthetic biology. And it should be a very interesting presentation. Our presenter today is Assistant Professor Reza Zadegan, and his research areas include nucleic acid memory, DNA nanotechnology, semiconductor synthetic biology. And he is actively involved in and focused on employing nucleic acid molecules to study biological processes, structural properties, programming, and informatics. His immediate one of his immediate goals, is integrating these concepts of nucleic acid memory and chromatin analogous gene expression, which he's used as CAGE and NAM, integrating these concepts to perform on-command genetic regulation to diversify biological and non-biological applications. So it's a pleasure to have you here, and we look forward to your talk.
Thank you, Dr. Murray. And hello, everyone. It's a pleasure to be here and present in front of you guys. Hopefully, you'll get information that is not usually available out there, because what my lab is focused on is at the intersection of engineering, specifically nanoengineering using nanomaterials, and biology, which is pretty unconventional. So as like Murray mentioned, and the title of my presentation today implies, we're going to talk about the unconventional applications of DNA. And if you're a biologist, usually you would think that DNA is just a genetic material. So you have cells that have genetic information, and then biology uses that genetic information and does what biology does. However, when I look at DNA, I look at it as a material. So for those of you, I think the majority of you that are chemistry major, we're looking at DNA as a material, as a chemical molecule, and we use it for applications that are not necessarily biology. Although there are examples, and I'm going to show you some of the examples of the work that we do in medicine and biology, but not necessarily all of the applications of the DNA that we're making or we're using is biology or biological. So when I start thinking about DNA, as I mentioned, I look at it as a material. And so one good example of why we need to look at material and try to program them. I remember that my lab is doing engineering with a material that is specifically DNA, right? So one good example of why we benefit always from looking at materials and trying to program them to try to use them in a more efficient way, usually is this image that I'm showing here on this slide. So if you look at the right and left image, both of them, hopefully you immediately get that, are showing mobile phones, and on the left side, of course, there are two strong men that are carrying a very old type phone. This image is from 1870, 80, around 100, 200 years ago. And those guys are carrying the mobile phones. But those mobile phones could only connect point A to point B, which was not that far, probably a couple of miles. And that was the only function that that phone had. But on the other side, you see my son, when he was 3, is holding a phone, and that phone is it's pretty old now. You wouldn't buy it anymore, but that phone has a lot of capacity. It not only connects point A to point B, which can be as far as a country, as far as a continent, uh, and that far, but also it can take a lot of images. It can record a lot of information. You can store a lot of information in that, right? So you can do a lot of computation that 20, 30 years ago, maybe 40 years ago, you needed a big building to do those computation. We have gone this far from uh, um, very primitive technology to a bit more advanced technology. And majority of that is because our community, and especially the semiconductor community, has been looking for programmable materials that specifically could conduct electricity, could do all those activities that we need in electronics, right? And that's why I think uh, looking into materials that are programmable is very interesting. Now, the next step of this revolution, I think, is going to be about big data and medicine. And today we're going to touch base on how we use unconventional materials such as DNA for these applications. We use DNA for storing information, and on the other hand, we can also use DNA to uh, do what I call intelligent medicine. And we're going to talk about that. So these are the future revolutions, the future, mostly futuristic technologies that we're looking at. And hopefully today, the conversation that we're going to have together. And by the way, feel free to interject, and if you have questions or if you want to talk about any of the specific things that I'm mentioning here today, feel free to interject. Either type in the chat or raise your hand or unmute yourself and talk about it. Anyhow, so today's focus is, of course, about storing information and securing information using DNA, and also in a very 180-degree, usually when we look at the science, right? So 180-degree different approach for medicine, for biology. So now let's start with the function of DNA that we know. And this is not a surprise, but sometimes when we hear this, it comes as a surprise because this is very, very well known, but yet undermined concept. The fact that DNA stores information. So DNA has been traditionally carrying information from one generation to another generation. This is the function that DNA has. If you look at the biology, what we call central dogma, the information comes from DNA. That DNA is a genome that you receive, every species receives from their ancestors. That carries a lot of information from the previous generation. There will be some changes, but usually that DNA is pretty reserved. That's a hard drive of a cell that you copy paste between generations, right? And what happens later is similar to what we have in our computer, that the hard drive has information, and then there is a processor that takes that information and processes it. The same thing happens in the cell. Through a process that we call transcription, RNA is being produced out of DNA. These are very temporary molecules that inform production of materials upstream that would inform biology. So that's what traditionally DNA has been doing, storing biological information. Now, we looked at it and we thought that, hey, how about storing digital data on DNA? And the question that you may have is, why would we want to store information on DNA? Where right now we're storing information on silicon, there is no problem. We have a lot of silicon everywhere. You can refine silicon without any problem. It's fairly available technology, right? The issue is that we have calculated with the rate of information generation that we have in the world, and you can relate with that some of you probably don't remember, majority probably don't remember, but 20, 30 years ago when we wanted to take a photo, we needed a camera, dedicated camera, and dedicated material to record images. But today, your phone has that or gives you that capability, and you go out and you take a lot of images that sometimes we don't use them, right? But we store those data. So that's a lot of information that we're just storing. And with the calculations that we have done, we believe by 2040, we're going to run out of space. So we're looking for alternative materials. That's why DNA might be that alternative material. You want to read more about it? Please read this paper that we published in 2016. It's about that concept. But over there in this paper, we prove that DNA has better energetics. We're talking about 7, 8 orders of magnitude better energetics. In another word, if today we store information and let's say a Google server farm, one of them uses or let's say all of those server farms, they use something like 40 power plant energy, or they consume that much energy. DNA can shrink that much energy consumption to basically 1 unit, right? So that's one big advantage. The other thing is that DNA has this capacity that you cannot see in other molecules, especially or other materials, especially that DNA has a resolution of about 0.5 nanometer, right? So that is something that we haven't yet achieved. Maybe in 5 to 10 years from now, we would achieve that, but we still believe that three dimension or a third dimension that DNA brings to the table, because it's a in solution molecule, would add a lot to the dimension. So we're talking about then a better density, about 1,000 to 10,000 times better density. And one other specific feature of DNA is that it can survive for a long time. You have heard about the fossils that we take out of soil, and we take the genome of that fossil out, and we can sequence basically a species that has lived thousands and sometimes millions of years ago. We're talking about fossils of, for example, dinosaurs, right? So it can survive for a long time if you give it the right condition, which is not really something that you can find in silicon-based material, because silicon-based material, majority of the technologies that we have, all the best ones, the state of the art, they can survive, the information on those can survive about 25, 30 years. We're talking about, again, state of the art material. DNA has hundreds of years of longevity. So that's another advantage of DNA. I'm going to skip through the rest of my slide here, but just quickly look at that single dot that my pointer is at. Probably very hard to look at, but according to a calculation that we had a few years back, that much DNA can store equivalent of 42 billion USB sticks. That well, I'm trying to give you a comparison between silicon and DNA. And of course, things have changed. These numbers have changed, but still, DNA provides that superior capacity. Now, to store that information in DNA, it's fairly easy. We're going to start with the binary data. You see it on the left side. You encode that into DNA. You synthesize those DNA molecules. These are fairly simple things. We'll touch base on how we do that synthesis, which is a pretty basic chemistry. And then after the synthesis, we can store it when we want to write or read the information. That's basically our write. But when we want to read the information, we just do sequencing. These are available technology, fairly fast, fairly easy these days. And then we can do some coding magic on it, But that's basically how you store the information. You just have a map and you store that digital information on DNA using that map, right? It's a very similar process to anything that we have in computers, right? Right now, I'm sending my video to you guys through the internet, and this is basically the same process. We have a map, takes the information which is mostly zero ones, right, and then sends it through the internet, and then you receive it, and your computer decodes that information back. Now, we have different ways of coding, but that doesn't matter. At the end of the day, you just convert binary, and DNA provides that quaternary. So now it means you're not only limited to that 0, 1, you can use the A, T, C, and G, which are 4 letters, right? If you are interested, we can talk more about it. But anyhow, this has become a bit more interesting, and in the last few days. I think since 2012, a few scientists have started working on this. There are some companies that have started working on that, developing different aspects of this technology. But there is actually a catch in the whole of this story. There is a video that you can right now go into YouTube and learn more about how DNA has that capacity, and you can store information on DNA. Just go to YouTube, search for the letters that you see on the screen. But this is essentially around 5 minutes video with, let's say, with 1080 resolution, right? If I want to store that information on DNA, which is the promise of that DNA storage technology, right? It would be very expensive. It will be around $5 million. These are calculations that probably changed during this last year of high inflation. But anyhow, around 5 million, $7 million. It uses a lot of energy, about 100 kilowatts of energy. It takes roughly about 4 days today, which is a very fast process for a biological process. Comparing it when let's say around 2000, this would have probably been around 4 years or 40 years maybe. So we have progressed a lot. But another aspect is that we're producing a lot of toxic waste, and this is essentially because we're using majority of the methods that we are using to synthesize that DNA is based on phosphoramidite chemistry. Phosphoramidite chemistry is very so it produces a lot of toxic waste, right? So, and we for that video alone, we will produce something around 15 liters of toxic waste, which is a lot, right? Now, so we believe that all of those issues can be solved. For example, the cost and the latency can be reduced by decreasing the volume, increasing the channels that we synthesize, and increasing the capacity of synthesis. We can reduce probably the power consumption. But the chemistry is still the same, so probably we cannot change that unless we are changing our approach. Just to give you an example of how disastrous this could be, if we store all of the information that we have in the whole world, these are just calculations. These change daily, but by 2025, let's say we have 1.4 bytes of the information. That would lead to around 15 petaliters of hazardous waste production, which is equal to the amount of water that Mississippi River puts into Gulf of Mexico, not for 1 second, not for 1 day or 1 second, but for 40 years constantly. That's gonna destroy the earth. So we're solving the problem of storage, but we're producing a problem. Now, we came with this idea of how about, no, don't synthesize DNA de novo, because that de novo synthesis is based on phosphoramidite chemistry. Let's take a DNA that is already existing And that we can take from biological material that we can synthesize in a green way, and then write information in that. And this is very similar to what you have in your computer, what we have in old CDs or in flash drive. You have an empty drive that you write information on top of that empty. When we say empty, it's in the state, the original state. You would just change the state of each of these bits from 0 to 1 and in a coded pattern, and that way you're storing information. So we had the same idea. Let's use a blank DNA tape and then write information or punch information on top of that. We use some biological magic. We mutate that DNA, and well, specifically in this work, we have mutated specific domains of DNA, and that way we write information. For example, in this case, we have a blank DNA, so starting with the top sequence, and then we try to mutate those domains. And when we read that information using our sequencing method, we would be able to relate the mutation to the data. So that's how we write the information. It's very similar to punch cards that we had old days. We call this actually a typewriter. So punchers in the old days when you had, you probably don't remember, but there has been a time in the history that music was based on these punch cards. I haven't seen them too, but I have seen videos about them. So punch cards had information about the music. You would put that into machine. It was I guess a piano. I guess there are some still around, and it was playing that music, right? So it's a very similar situation. You punch information on top of the DNA. In our hard drive is very similar. The hard drive reads by changing the state of each of those bits. It punches the information. So that's what we have done using the technology that we use to punch those information on top of DNA is via the very well-known CRISPR. So we know that CRISPR can modify DNA, right? So we use that DNA, that CRISPR to modify our states, our domains from one state to another state. And we can just easily guide them because CRISPR uses a guide RNA. You can just specify the specific location that you want to modify. And then to read that, we just need to do the computational magic, right? So all of that we do in our lab. And this is an example of the data that we've written on DNA. These are exactly what we have done in the lab on a real DNA. We had the title of our paper. We encoded that into the punches that we wanted to put on top of the DNA molecules. We had a few registers, wrote information on it, and then we recovered the data back. If you are interested, we can talk more about it. That paper was submitted, and you can find it in PubMed using that ID and the OI that is over there. So if you're interested, please read that. But anyhow, there is a need for connecting this with the real computers and try to make these as similar as possible to the hard drives. We use microfluidics. This is a paper by my student Hassan that did the microfluidics for this project. I'm gonna skip through that because I think we're probably, are we running out of time? Do we have?
No, we are good still. Another 15
okay.
minutes or so.
Yeah. 15 minutes. Okay, sounds good then.
Mm-hmm.
All right. Yeah. So the second type of application that we get from DNA is this is a bit more fancy, I would say, because DNA has been, the material of information for information storage forever. But this other application that we get from DNA is a bit fancier. So we're trying to make structures using DNA molecules. We call these DNA nanostructures. We're talking about very small structures. Comparing an apple to the earth would be the comparison of those structures that we make to the apple. So we're talking about a very, very tiny structures. It's fairly simple. One of the examples is that if we have a long strand of DNA, we just define the complementary strands and we design this. This is a process that you can easily do. I'll show you how very quickly. You do have those other DNA strands. It forces that longer DNA strand to take that specific shape. These are all artificial, remember. And then we can lower the temperature, the chemistry, and the thermodynamics. So it will force that longer strand of DNA to take the shape that we want. And we can design these very deliberately. And as you can see here, I have examples, a variety of different examples that we have been doing in our community. We can make 2D, 3D structures. You can see that we have a painting of Mona Lisa, not very similar to the original, but pretty close. We can make smileys, right? And the size of them is very small. Again, we can control that size. I've made the structures that have been smaller than a ribosome and the structures that have been a bit bigger than ribosome. But generally, we're talking about between 10 to 100 nanometers, or sometimes a few hundred nanometers. To do that, we sometimes can do all of these manually, but it takes really long time and it's a very, very laborious and error-prone process. We have developed a software. You can find that software from our GitHub repository, and it's fairly easy. It takes a few seconds, as you see here. My student was able to write and basically make this very complicated structure in less than 25 seconds, less than 30 seconds. And so we develop these, and then we use the software. We buy these DNA molecules. These are small strands of DNA. We mix them in a pot, and then AFM imaging them shows that these structures usually form. This is a data that hopefully we will publish soon, but so there are indications that the structures that we made were forming. It's not very clear in these AFM images. Our AFM, we've just switched our AFM. This is not the best AFM that we could have, and not necessarily the best images. These are improved. This is a bit older data, but you can see, for example, that ANT we were able to make many different shapes and structures. Now, the question probably that comes to your mind is, why would we bother to do this? There are different applications. I'll show you medical applications later. There is one that I specifically want to show you guys, but you can imagine that using these structures, we might be able to solve the the issue of the top to down production, which when we need to basically shave materials and shape them in a specific shape that we want. With DNA, we do the bottom up, so it would be chemistry wise, it will be faster and more efficient because you have molecules that work for you instead of one equipment that punches the material. And these have different applications. Could be in electronics, for example. And we can make switches and different types of materials. Examples are the one that I'm showing here. We can also write information on top of these structures. This is an example that we recently published, a paper on writing that sentence, DNA data is in our DNA, on top of these origami DNA structures that we make. So this is very similar to that punching concept that I was talking about. We use super resolution microscopy to read excuse me, to read back the data, and we showed that we could write the information on top of these structures. So it's basically performing as a breadboard to write information. We can use these for patterning of material in electronics as well. Now, Another application of these is in mostly biological and medical applications. Um, uh, so look at this slide. Basically, this summarizes everything. But uh, please memorize that part that I have in the middle, which is two inputs with a logic gate produce output. This is just an AND gate, right? So we have two molecules reacting with each other, two DNA molecules reacting with each other, producing the third molecule, simple AND gate. So now the question is, that comes to your mind as, what is the relevance, right? So we can make these type of logic gates. That's the message. Now, the question that comes to your mind probably is why logic is important. Logic gates are important, especially in biology, right? Look at this metabolic pathway of E. coli. And if you can read it in, in let's say 1 hour, I'll give you a prize. Just kidding. Probably I won't, but this is very complicated, right? It's possible to read it. It's very complicated though. Now, this is the language of cell. This is how cells communicate with each other. You see on the right side of this image, there are a lot of outputs and inputs that cell has with the environment. There are a lot of communications inside the cell. It's very complicated. You cannot easily read this. But if I focus on a very specific location and look at it, so the language may seem a bit hard to understand, right? But if I look at one specific location, let's say, for example, like opera, I can look at that and see, oh, this is basically a logic gate, nothing more than a logic gate, right? Like opera, you have glucose. Basically you have glucose. It will perform. Now, you don't have glucose, which is very important for survival of the bacteria, doesn't have the food. Now, it should do something. The bacteria is going to use this like operon. If you don't have glucose, then there will be a protein produced that's called CAP. And if you have lactose in the environment, now bacteria cells that prefer glucose as food because it's much faster, much easier to process, they use lactose as their food for their survival, and that becomes a trigger of production of or activity of beta-galactosidase enzyme, and as a result, the bacteria can use lactose as a food. So it's basically a NOT gate and AND gate together, right? Not glucose and lactose. So language of cell. This message here is the language of cell is logic. And now I talked about, hey, we can use these as logic performance, right? So now we know the language of cell. We know that there's probably a chance that we can play with that and speak that language. I just need to show that I can speak that language. We have made structures that these perform logic in reality. Say we have a structure that's made out of DNA. I'm not going to go into detail of this, but this is a box structure. And we'll talk about why box, but this is a box-shaped structure that slit can be open or closed based on the gate, the inputs that we give that gate. This is basically a logic performer. For example, here we have an AND gate. This takes two of the breast cancer microRNAs. If they are existing in the solution, in the environment, the box is going to be open. You can imagine where I'm going. If I have a drug inside that cage that is basically packed, and this goes to a cancer cell, which is breast cancer cell, let's say, those microRNAs that are only existing in those cancer cells open up these cage, the drug becomes available, the cell is going to die. What is the advantage? Compare that to the chemotherapy that we have right now, which is basically affecting everywhere. It goes to all the cells, it affects all the cells. You would have a lot of side effects. Those are the effects of drugs nowadays, right? So we're talking about something that is a bit smarter. It goes to a specific cells. And even if it doesn't go to a specific cell, those markers only exist in that specific cell that we're targeting. As a result, it would not react in normal cells. So you're not going to have any damage to the normal cell. Uh, um, we can imagine that if I have, let's say, an enzyme that reacts, or a substrate that reacts with my, if I have my substrate basically inside the cage, if I don't have the key, it will be closed. In this example, we have an enzyme that doesn't have access to the substrate. When we don't have the keys, when we have the keys, it will open up, those keys open up the lid of the cage, the structure, and as a result, the enzyme has access to the substrate. That substrate can be released. That substrate might be a drug. You get the story. We will solve the problem of intelligent medicine. Now, we went one step further. So we showed that we can do that, right? But I don't have the data here. You can find that data in my papers. But anyhow, we also wanted to go one step further and now express a specific gene. We wanted to show that we can even control genes using the same technology. In a technology that we call CAGE, that stands for Chromatin Analogous Gene Expression. We kind of borrow everything from cells and how they behave in nature, that their promoters are pretty packed, not available. By giving the availability to that promoter, we would be able to shut down or turn on the gene expression. So basically, again, we can have the markers that react with that structure, and if this structure is active, you can imagine that we can produce that RNA that is coming from that gene, right? And that gene could be for example, a necrosis pathway activator. So you would imagine if I have these structures inside a cancer cell, I can express them specifically in those cancer cells and kill the cells. That's the whole idea. We do have some ongoing experiments, and hopefully we'll have much more to show you later. But with that, I think I will wrap. I would like to thank all the people that have been helping in making these research possible, especially my collaborators, my lab members, and also the agencies that have supported these works so far. And with that, if you have any question, please just ask. I'd be happy to answer them.
Okay, thank you very much. Very good. So any questions from anyone? You could unmute.
Yeah, one thing that I forgot to mention is that we're looking for students and postdocs, and also before that I mentioned that we're also looking for faculty. So any of these, if you have any interest, if you know someone that has interest in doing that, please just reach out. The information that I have here is up to date, so you can just send email, go to our website, or call me. We are graduate only program, and we support the students. So keep that in mind if you want to. Meaning that you're going to be not only supported, but also compensated for your activities that you do in the lab if you decide to join. Yeah. But with that, please go ahead with the questions.
So okay, so you just said, and I know that you are a graduate program, but do you have summer research opportunities though for undergrads?
We do. Okay. Actually, I am leading an REU for synthetic biology this year. So if you're interested in it's possible that we prefer in person, but if someone for any reason has hardship, it is possible to do it online as well. Just
Mm,
reach out. We do have the opportunity for um, uh, research engagement for the undergrads.
for undergrad.
Every year, specifically this year, I'm leading that effort. Just reach
Okay.
out.
All right. That sounds good. And I was wondering if you are considering applications, several applications, like in making DNA molecular machines. Is that one of the things
Uh,
that mhm.
can you clarify the question? I think I missed the
Yes,
part.
are you thinking about creating molecular machines from DNA?
Oh, okay. Yeah, yeah. So these basically these materials that I'm showing, specifically this one, these are machines. Now,
Okay,
if you're looking for moving these, so these are structures that are made out of DNA, and we can switch their state, meaning that they
okay.
kind of perform as switches and machines. But if
See,
you're looking for um, things that move, there are a lot of research in the community that has been focused on that. We do have some interest in that as well. However, our approach is mostly using these as a vehicle to control gene expression nowadays. So most of our applications are limited in that. But yeah, absolutely, we can design and make those structures. If you're interested, we can talk more about it. We can definitely do that.
okay.
One thing that you can do is to go and use this code for your designs,
Mhm.
um, and define modifications that you need for making this structure to move. What I have actually here on the very bottom of the this slide on the left side is an arm, a robotic arm that is made out
Mm.
of DNA, right? So that's
Okay,
supposed to move around.
okay, okay.
Yeah.
All right. Other questions? Just unmute yourself if you have a question.
Yeah, Professor John Rohrbeck here. I can almost picture being able to store stuff onto DNA. I'm just curious, is a biological system like that going to be robust enough for storage? It's probably a whole different unit working on storage questions, but that I don't see how you could keep that stable.
Very good question, and this is all the time on our minds when we're looking at this system. And basically, one thing that I probably did not mention, or not it was not clear, is that we look at DNA as a non-bio well, for our application, non-biological material, meaning that it will not mean anything to biology, and we don't actually want any biological activity from these molecules that we synthesize, and we keep them out of biological systems. That's a good question. There has been works that people have tried to store information in cells, for example. I generally don't think that's a good approach or a viable approach for many reasons. First, the information in biology needs to be fluid, although the rate of mutation in biology is very low, right? So it takes millions of years to change the information. But that's how evolution works, right? But that should happen. The system is designed so that you get the evolution. So, in another word, the data should not be stable, right? Or at least should not be stable for forever. Um, However, I think for all those complications that comes with the like putting these in cells, and on the other hand, not really much gain that you get from, because there's basically no advantage of storing information in cells. Uh, uh, not a uh, very convincing argument. So as a result, I don't think that that would be a problem. On the other hand, when you store it in laboratory condition, we can, we have shown actually in that paper, we have calculated and we have some evidences that shows DNA can survive for hundreds to millions of years given the right condition, including low temperature, probably no ionizing waves and no ionizing radiations, and at the same time probably not having much oxygen or water around would be the solution to make sure that DNA can survive for millions of years. Now, if I dry that DNA and keep it on room temperature, I guarantee that it can survive for hundreds of years. So that's the stability that we're talking about. Now, you can improve that, of course, right, by those things that I mentioned. But I don't think really in biology is the viable solution to go for. Hopefully I answered your question.
So then you would probably hesitate to use, your approach to make antivirals or antibacterials because they would have to interact with, for example, human cells?
That's a good question. Well, now these systems that we develop, we can guarantee that there will be no communication with specific genome or specific cells. Let's say, for example, in prokaryotics and eukaryotics, they have different promoters, right? Mm-hmm. So for that last example, the cage example that I had, I can imagine that I can specifically mention to this structure or indicate or ask the structure to be very only respond to bacterial promoters, basically, or bacterial enzymes. So as a result, it would not really do anything in a eukaryotic biology, right? So as a result, I don't think that would be a problem. So that's possible. That's
possible. That's okay.
Yeah.
Okay. All right. Other questions? Anyone? Okay. Well, if there are no more questions,
I'll ask one question here.
What
could you go back to the slides on some of the error rates in writing and stuff, and like just maybe say a little bit more about?
I think you're talking.
Yeah, right there. Yeah. I thought that was that's pretty interesting. I mean, how that's probably got to improve, but it looks like it's been improving greatly. As you're more, maybe you could just say a little bit more.
Sure.
How well that is working in terms of error rates and stuff.
Sure. One thing to keep in mind is that what I'm representing here can change by changing the system and design.
Sure.
So these numbers by no means are going to be exactly the same if you have a different
for sure.
system. But anyhow, what we have done was to punch the information on top of that DNA that we have, right? We have different registers. These registers are strands of DNA, individual strands of DNA that have different domains on them. And the writing of the information is basically transferring that binary data, which we get from, for example, here we have the title, which is in text, which we use ASCII to binary. We convert that to binary. Now we want to write that on top of the DNA, the several registers that we have. Now we have written a code that does those calculations for us and tells us exactly where we need to add which guide RNA, which system we need to add to specifically modify that domain, right? In a specific register. Now, after you do that, that's the storage. That's writing, and right. We're stopping at that when we say just the storage. So that's the storage. Now, when we want to read it, we do sequencing. And sequencing is a progress, right? It's a process, and it's a progressive process, meaning that you read more, and as you read more, you would be able to get more information because you're uncovering those parts that are not being read. And during this process, of course, depending on the type of sequencing technology that you're using, here we're using nanopore sequencing, which is error prone comparing to, for example, Illumina sequencing. But depending on that sequencing technology that you're using, you're going to get some errors. And remember, when we were writing the information on top of the DNA, we added, I think I didn't mention, but we added some error correction codes, which means we should be able to recover the data after we have some majority of data. Because we have the error correction, right? And in this specific graph, what I'm showing is actually that process. Now, we do have a raw reads and errors that we get from them, right? And as we have more reads, the errors go down drastically because it's a collective activity.
Oh, okay.
Or if we can call it collective activity. These are examples of 100 read. We don't have coverage of all of these registers. 1K, 1,000 reads, we have coverage of some of the registers, not all of them still. In 10K, we do have majority, but still a lot of errors. And with 100K, the error was still high to some extent, but we have majority coverage. Now, depending on the code that we have written before, we can stop at any of these steps and recover the data. It depends on how much overhead we pay in when we're writing that code. Now, for this specific work, we have written it such a way that anything below 30 errors would be recoverable.
Oh, okay. Gotcha.
It happens that 100K would be at that time that we will be able to recover the whole data as one block of information, intact block of information. And that's why we have no read, no basically recovery, and it's all false until around 100,000 reads for this code, right? Which suddenly it is recovered. The whole information, as the whole information with 100% integrity is recoverable.
Now, I tell you, the biological, think of it, how to read DNA biologically and then mapping that with, error rates and reading, digital code. I think that's what the problem I'm having translating. But once you get enough of the code, you can, I'm understanding now that you can get all the code. You're not actually using it. So
yeah, exactly.
So that's a weird thing about DNA. Maybe that's an advantage then.
Yeah, well, so one advantage of DNA, which we don't actually use in this specific work, but one advantage of DNA is that collective information that it has, right? So you don't have one molecule of DNA, you have several molecules of DNA. We're not taking advantage of that in this specific work, but you can imagine that if I have 1,000 DNA molecules that have the same message, right? I can recover the information faster because there is a bigger chance that I get more of that message coming from the pool that is bigger. Now, of course, it's a computational problem. And in biology, it's very similar to the genome processing. So when you do genome reading, for example, you want to do the whole genome of a species, you would have a lot of reads, and then you try to assemble them together and get that information, that consensus information. It's kind of very similar, but what we call majority voting can be a very advantageous situation. Actually, to some extent, you see that majority voting. The majority voting shows up in our the chart that we have here in our raw data, right? I'm not sure if you see my mouse, but
I see it.
That's where I have the majority voting helping me. But at some point, majority voting is not gonna improve data recovery anymore, or not with the rate that we're satisfied. we can read 1 million times, 10 million times. It would probably take a long time, a lot of expense, right? So that's where you put that info, that code. And it's just arbitrary. I can choose to put that at 5K. I can choose to put that at 200K. Whatever I want, I can play with. There is a lower limit. We're trying to figure out what that lower limit is, but basically, as much as I am around this area, more than 60%, I should be able to recover the data if I have enough overhead coming from the error correction. Hopefully that kind of solves.
Yeah, that helped me get a better picture for it. Thank you very much.
Good question.
What kind of background would students, I feel like you got, computer programming, you've got the biological parts. What kind of background would really help students kind of jump into this research?
Yeah, well, anything, but of course relevant, right? You wouldn't probably need to. I mean, I do have students that don't know even coding. It's just that I do have probably a couple of students that have never coded. They probably now want to learn some of that because that helps communication. I do have a student that was a electronics major, and I have a student that was a computational only person, meaning that only writing code. That's the only thing. I have that team. So everyone together, we have a chemist, we have a biotechnologist, we have a biochemist, we have a physiologist, we have a molecular biologist. I have all of these different people in my team. I believe for what we're doing, diversity always improves our work. Anyone that comes to the team brings their own appetite, makes the dish much more delicious. That's my way of looking at it.
Nice.
Yes.
Yeah, that's very collaborative. I love that's a great way to say that and share that. Bring what you have and let's see what it could contribute. That's very nice. Thank you.
And there's always a place for everyone, right?
Yeah.
Yes. Okay, so thank you again very much, Professor Zadegan.
No problem.
We will stay in touch. Yes, we will stay in touch.
Absolutely.
And perhaps one or two of our students may apply. So do you have a deadline or it's for application for summer?
So if you go, I can send
it to me.
I don't have it on top of my head, but I can send the information regarding our program. Right now we're accepting applications, and there will be a need for a recommendation letter or a couple of them, and so it's better if the students start as
doing it now,
as soon as possible. But right now we're accepting the applications. It's a summer program, so we want to wrap it up or wrap our assessment of the students before we choose the students, probably by mid-April or before that.
Yeah, that sounds reasonable. All right, thanks again very much.
Absolutely.
Say hi to Shireen for me.
Absolutely, yeah. Thank you.
Okay, all right.
It was good to thank you for having me. It was good to see you guys and talk to you guys.
Thanks for being with us.
Have a good one.
Okay.
Thank you.
Bye-bye.